Welcome back, everyone. We are here for session number 3. This is the Trainee Showcase. A little bit of a shorter format. Each speaker will spend 5 minutes presenting. We won't get into question period after each speaker. Instead, we'll let all 4 speak right through, and then we will have a panel discussion. So still feel free to post your questions in the question and answer forum box, and I will get to those and direct them to each speaker at that time after we've heard from everybody. I would like to introduce our first speaker, Dr. Yoshi Kubo. He is you're a postdoctoral fellow at the University of Lethbridge.
Yep.
You received your PhD in computer science from Dalhousie University, and his research area is bioplausible deep neural networks and adversarial examples. Yoshi, I hand it over to you.
Thank you. So, can I start it? Yeah. Okay, let me start. Thank you so much for introduction. I'm Yoshimasa Kubo. So, I will discuss biologically inspired neural networks. Next slide, please. So, we have seen that many artificial neural networks solved many problems, such as game of Go. So, world top player of game of Go was defeated by artificial neural networks that is trained by backpropagation. But actually, backpropagation is not biologically plausible. Next slide, please. So artificial neural network trained by backpropagation does not solve all the problems that humans can solve, such as multitasking. For example, you can listening to music while you guys are studying, right? Another example is adversarial examples. So here is an image of a panda, and we can classify it as panda, right? But artificial neural network cannot classify it as panda, but gibbon, a monkey, with 99.8% confidence. I know it is a little bit weird, but it happens to neural networks. Next slide, please. So, of course, there are some biologically inspired training algorithms, such as constructive Hebbian learning, CHL, equilibrium propagation, EP, difference target propagation, DTP. These are based on STDP training algorithms, but these are not powerful enough like backpropagation and our brain. Next slide, please. So to create powerful algorithms like backpropagation, I think more than backpropagation, our brain. So we still missing some parts for biologically inspired training algorithms. So we would like to find these missing parts for biologically inspired neural net training algorithms. Then we would like to solve as many problems as possible. So next slide, please. So what we have done so far is, first one is, recently we have published a paper on nature machine intelligence. So in this study, we have applied a hypothesis to biologically inspired training algorithms, such as CHL and the EP. So this hypothesis is a single neuron predicts unsteady activity. So we applied this hypothesis to the CHL and EP and trained on the image classification tasks. And these worked well. And another study is, next slide, please. So neural adaptations. So neural adaptation can be seen in our brains and many systems. And then we applied a neural adaptation to constructive Hebbian learning. But this constructive Hebbian learning is very limited, such as hidden layer is very small and dataset is too small. So here is a question. Next slide, please. Does neural adaptation work in more complicated models and more data too? To answer this question, we apply the neural adaptation to constructive learning and EP. These are more complicated, and EP is actually convolutional neural network, deeper model. Next slide, please. So actually, yes, it works well. They worked well, actually. So in this table, no adapt means without neural networks without adaptation. And adapt means neural network with adaptation. So we applied these several architecture to the MNIST and the CFAR 10 dataset image classification tasks. And then we checked the testing error and the training error. And the bold ones are most lowest error. So error means lower is better. So bold one is the best ones. So we could see that our model with adaptation works well. So hopefully we will publish this paper sometime soon, and then you guys will enjoy this paper. Thank you so much for listening.
Yoshi, thank you so much. Really appreciate it. And Yoshi will join us again once we get back to the panel in a couple more minutes. Our next speaker, I would like to introduce Dr. Maria Masoliver. Maria did her bachelor's and master's in physics at the Autonomous University of Barcelona and both Free University of Berlin and at the Technical University of Berlin, respectively. She specialized in complex systems, more specifically neuronal dynamics. This led Maria to the Polytechnic University at Catalunya for a PhD in computational physics, where she tried to understand how neurons encode a weak signal in a noisy environment. Maria, thank you for joining us.
Thank you for having me. So welcome everybody. Today I'll talk about embed chimeras in recurrent neural networks. Currently, I'm a postdoc at the University of Calgary in the Physics and Astronomy group under the supervision of Pilte Nicola and Jörg Davidsen. Next slide. So what is a chimera? In a chimera we can find synchronized and asynchronized activity. Next slide. And actually you can click also again. Yes. So in a chimera, as I said, we can find coexisting domains of synchronized and asynchronized activity. And chimeras can be found experimentally in couple metronomes. As we can see in this figure, each little blue dot represents a metronome. And we observe that we have two different behaviors, synchronized and asynchronized. They are all coupled together and they are equal. So this was actually a surprising result. So different behaviors were observed in equal oscillators. Next slide. Chimeras have been related to epileptic seizure onset. As we can see in this example, where the electrical activity of a patient with epilepsy is plotted, each little dot is the phase of several neurons measured by an electrode in the scalp. And we can see that before the seizure, we observe a chimera-like behavior because we see synchronized and asynchronized activity. Whereas after the seizure, the electrical activity synchronizes. Next slide. So why are chimeras important? So as I have just said, chimeras can be related to brain disorders such as epileptic seizures. So studying chimeras can shed light on the mechanism underlying seizure onset. Next slide. The particular aim of the project that I'm working on is to understand if chimeras can emerge in neural networks. That is to say, how in the macro scale, so how the small neurons, the neurons arrange themselves so that in the mesoscale, let's say the brain regions, we observe a chimera. In this case, the epileptic seizure. Next slide. In order to do that, to answer this question, we will train a recurrent neural network to learn a chimera state using the fourth method, which is a machine learning approach. Next slide. Here, I want to point out that when I say network output, it means chimera state. Next slide. The method that I'm going to use, as a lot of machine learning techniques, has 3 steps. We have the pre-learning, the learning, and the post-learning. Next slide. And actually, you can also click. Yes. So in order to explain you about this, I'll present this simple example of learning a sinusoidal signal. And we start with a sparse matrix, which we observe that has chaotic activity. Next slide. Let me jump from the pre-learning to the post-learning, where we added a second set of weights that learn the activity that we want the network to perform. And we see that the firing rates are regular. Next slide. From those firing rates, we can get the network output. So it's actually a sum of the firing rates that give us the network output. Next slide. And we can see that is equal to the supervisor. The question is, how has the network learned? So next slide. At each time step during the learning period, the network output is compared to the supervisor. This error is fed back to the network, and the weights are adjusted so the error is minimized. If you want to know more about it, just check it out, this reference that is here. Next slide. So the supervisor that I'm going to use is the chimera state, the one that you can see here. And it has 6 metronomes coupled between each other. And we can see that half of the metronomes are synchronized and the other half are not. Next slide. Here is the dynamics. Again, we can see the time series of the metronomes, synchronized and not synchronized. Next slide. So we can see that the recurrent neural network learns the chimera state. The black line represents the supervisor, whereas the colored line represents the network output. Next slide. And interestingly, from the firing rates, we can obtain the information of the chimera state because we perform a fast Fourier transform of the mean of the firing rates, and we obtain the frequencies of the chimera state. Next slide. So as a take-home message, chimeras can emerge at the mesoscale and macroscale. Next slide. Which lead us to think that chimeras can have relevance in neuroscience, in pathological and healthy circuits. There were a lot of more other results, but I could not fit them. Just bear in mind that I will publish soon these results, so check me out. Thank you very much for your attention. Next slide. Yes.
I'm just having trouble getting my video on here. There we go, Maria. Thank you so much. Our next speaker is Tejal Aslesh. Tejal is a graduate student from Dr. Yoshifumi Yakota's lab at the University of Alberta. The focus of her research revolves around the development of novel antisense I'll let Tejal pronounce this.
Yeah, it's oligonucleotides.
There we go. I'm a meteorologist, and I sometimes can't even say meteorology. I stumble with that. So that is for the treatment of spinal muscular atrophy in model mice, known as SMA. Her research aims to address the issues associated with currently approved treatments for SMA. Besides SMA, the lab also works on other neuromuscular disorders, such as Duchenne muscular dystrophy and FOP. Tejal.
Okay, thank you for the introduction. Okay, hello everybody. My name is Tejal and I'm a 5th-year PhD student at the University of Alberta. Today I'll be telling you a little about what I work on, which is basically DNA-like band-aids for the treatment of SMA. Next slide. So spinal muscular atrophy, or SMA, is a genetic disorder that predominantly affects infants and children. It leads to progressive muscle weakness, making these patients lose ambulation. They're often paralyzed or they're wheelchair bound. This progressive muscle weakness also results in respiratory distress, making them rely on assisted or non-assisted ventilation techniques, which may be invasive or non-invasive. And unfortunately, this disease is the most common genetic disorder in infants. Next slide. So SMA is caused because of a mutation that leads to deletion of a gene called survival of motor neuron 1, or SMN1. This ultimately leads in degeneration of motor neurons that basically relay information from your central nervous system to the muscle. Next slide. So what are DNA-like band-aids? Let me give you an analogy. Imagine you cut your finger. You use a band-aid to stop the bleeding and to cover up the blemish. And imagine a cut or a mutation in your DNA. Here you use DNA-like band-aids or antisense oligonucleotides to cover the mutation to ensure proper functioning of the DNA. Next slide. So, healthy people have two copies of this gene. One is SMN1 that can produce 100% functional SMN protein, making motor neurons very healthy. They also possess a copy of called SMN2, but because of a tiny mutation in the DNA, it can produce only 10% functional SMN protein. Now, the problem arises in SMA patients because they do not have SMN1, and this 10% functional protein is not enough to compensate for SMN1 loss. So here comes the DNA-like band-aids into the picture, where they bind to the mutation present in SMN2 and increase the production of functional SMN, allowing SMA patients to live. Next slide. So one such DNA-like band-aid or antisense oligonucleotide that is approved for SMA is called nusinersen. So this drug cannot be systemically injected because it cannot reach the central nervous system tissues, namely the brain and the spinal cord, which is why people rely on lumbar punctures. So these injections are very, very painful and cumbersome, and they often lead to renal toxicity, bleeding complications, immune response, etc. Next slide. This brings us to the hypothesis of my project, which focuses on a non-invasive systemic delivery using a peptide conjugated DNA-like band-aid for SMA treatment. Next slide. Here we have a molecule of DNA, and on the right, we have a DNA-like analog called PMO. We conjugated it to a cell-penetrating peptide called DG9. So this peptide basically increases the uptake of our DNA-like band-aid in target tissues, and we can rely on systemic injections for treatment. Next slide. The experimental procedure for my project involves using a mouse model which has SMA. It exhibits similar characteristics as seen in patients, which includes muscle weakness, breathing difficulties, and these mice, they do not survive without any treatment. So we inject these newborn SMA mice with our DNA-like band-aid using a non-invasive subcutaneous injection into the skin, and we harvest tissues on day 7 post-injection. Next slide. What I do in the lab: post-dissection, I freeze the samples, we process the DNA, we use several staining techniques to assess the health of the muscle. We also look at the interaction of the brain and the spinal cord with the skeletal muscle. Next slide. Here is a glimpse of my results. We mainly wanted to see if our treatment prolongs the survival. For example, here, we have the non-treated mice here in orange, and these mice typically survive only for 8 days. And here in red, we have our DG9 PMO-treated mice, which have a median survival of 58 days. And on the right, we have a picture of our mice. As you can see, the DG9 PMO-treated mice look very, very healthy, just like our healthy control on the left. And on the extreme right, we have our non-treated mice that look extremely, extremely weak. Next slide. So in conclusions, next slide, we see that our treatment actually tremendously improved survival. It definitely increased the gene expression. We looked at functional testing in these mice. Obviously, I cannot share all the results in this presentation, but we observed really promising results following our treatment. Mainly, we bypassed invasive injections. Next slide. And I would like to thank my lab and all the funding agencies for their support. Thank you.
Thank you so much, Tejal. I'd like to introduce our next presenter, Dr. Joseph Kamtchum Tatuene. Dr. Joseph is a neurologist with expertise in neuroepidemiology, neurovascular ultrasound, and transitional stroke research. He has published over 50 research articles and reviews with major focus on biomarkers of stroke and carotid atherosclerosis. He is on the editorial board of the journal Stroke and Reviews for Stroke Neurology and the European Journal of Neurology. He has received awards and funding from the American Academy of Neurology, the Wellcome Trust, Alberta Innovates, and the Killam Trust, as well as the Bank of Montreal Financial Group. Welcome, Dr. Joseph. I'll hand over the stage to you.
Thank you very much. Can I have my slide? Good, thank you very much. So my research is what you just heard. Good afternoon. Focuses on finding new biomarkers and new therapeutic targets for patients with carotid atherosclerosis in order to improve the management of carotid atherosclerosis or the prevention of stroke. Next slide, please. So I don't have any financial disclosures for this presentation. Next. So carotid atherosclerosis is the cause of about 2 million ischemic strokes every year. When we talk about stroke, we really refer to blood clot blocking an artery in the brain and causing symptoms like weakness or difficulty to speak or to see. And interleukin-6, which is the biomarker of interest in this presentation, is just a protein that could be found in the blood and that has been shown to promote inflammation and deposition of cholesterol in the arteries, especially in the neck, what we call it carotid, as you can see on the right side of the slide. And these results really derived from animal studies. So our question here was, is it also possible to find a relationship between this interleukin-6, which is a product of the immune system, normally helping people to fight infections and various injuries, but is it possible to find a relationship between this interleukin-6 and the progression of carotid atherosclerosis? When we say progression, it's really the narrowing of the carotid atherosclerosis that becomes progressively worse, just limiting the blood flow from the heart to the brain. So this is important because we already have drugs targeting interleukin-6 in clinical practice, but they are used for other diseases like cancer and rheumatoid arthritis. Probably know about colchicine or methotrexate. And these drugs could be easily repurposed to improve stroke prevention in patients with carotid atherosclerosis if we demonstrate a relationship with carotid atherosclerosis. Next, please. So to investigate that question, we use data from the cardiovascular health study. These are publicly available data that were collected in the 90s in the United States. And in that study, what they did is a carotid ultrasound, as you can see on the right side. So it's just a regular ultrasound of the neck artery to measure the narrowing. If there is a plaque or cholesterol deposit, the narrowing, and score it from 0 to 5 according to the percent narrowing of the lumen. They also measured interleukin-6 in the blood of patients. And at 5 years, they saw those patients again and then measured the narrowing to see if there was any change, if the score has progressed from 0 to 1 or 0 to 3, depending on the patient. So what we did is apply some of the machine learning techniques that are out there. We call it logistic regression, to study the relationship between interleukin-6 in the blood and progression of carotid narrowing. Next slide, please. So the main result here, it's only one of the many results that we could present, is that patients who had a blood level of interleukin-6 greater than 2 picograms per milliliter had a 25% higher risk of having a progression of their carotid disease, that is increased narrowing of the carotid lumen when they were seen 5 years later, after 5 years after the enrollment. And this was independent of all the other factors that typically affect the progression of carotid plaques, especially hypertension, diabetes, or smoking. But we also saw an association or an increased risk of carotid narrowing related to those risk factors. For example, you can see here abnormal lipid profile is related to 129% increase in the risk of having a carotid atherosclerosis progression. And this progression is a risk factor for subsequent stroke. Next slide. So the main conclusion here is that we have an independent relationship between this interleukin-6 in the blood and the progression of carotid disease. And we need to have trials that will test this interleukin-6 drugs in patients with carotid atherosclerosis to see if we can prevent progression and onset of stroke. But while waiting for those trials, we still need to carefully manage all the cardiovascular risk factors and promote smoking cessation and healthy diet. Next slide. So with this, I would like to thank all the people who contributed to this work and the institutions that funded my research. Next slide. Thank you for your kind attention.
Thank you so much, Joseph. I'd like to invite everyone back onto the stage to do a 10-minute panel discussion. We have some questions that the audience would like to ask of you guys. Looks like everyone is back up. Excellent. Well, thank you so much for sharing your work with us. It's quite interesting. We have a question here for Tejal. We noticed that there were 2 different doses in your presentation, in your work. And is there a dose-related response that you've identified at this point? And also, have you tested longer than 7 days?
Yeah, so we did test 2 different doses and we observed a dose-dependent effect on a lot of things like gene expression, survival, and we did test them in older mice, at like 1 and 2 month old mice. So yeah, our treatment still prevails its effects at that time. So yeah.
Interesting. And your treatment, you were providing that treatment for greater than 7 days?
Yes.
Yeah, okay.
We just inject them once and we observe effects at different time points to see how long our treatment is actually efficacious.
Okay, so you're monitoring over a longer period of time.
Yes.
Excellent. Thank you. There's also a question here for Yoshi. Is the likelihood of neural nets matching the human brain's accuracy in identification by 2030?
It's a hard question, but maybe, so I'm not sure if I can answer this question, but I just hope so, because right now many people are trying to apply, some neural such as knowledge of the neuroscience to the training algorithm, the neural networks. And then hopefully we will solve the many problems such as, image classification task or, 100% classification accuracy or something like that.
Excellent. So you don't have a crystal ball?
No. Finger crossed.
Oh, thank you.
I'm going to hop in because there are some members of the public, some who may not be very familiar with research, with the intricacies of research. And so we can just open the discussion a little more broadly, talk about, we'll move through sort of in the same order we started, something that maybe the public would be unaware of that your group does in research, something that might surprise some of the folks from the public about your lab or something along your journey that is just an interesting tidbit. So let's start with Yoshi, Maria, Tejal, and then Joseph. Yoshi, we'll go with you first.
Okay.
Yeah. Go ahead.
Sorry. What did you say? Sorry, I couldn't hear it.
Okay. Let me pose it again. Is there something you can share with those in the public that are on the forum that might surprise them about your research, about your lab, about your team, just something in general, a challenge that you meet frequently in the lab, something sort of anecdotal that you could share with folks from the public?
Actually, so recently, I have not worked in the lab. You can see, I'm in apartment. So it's very hard to communicate with, people in the lab because we have to use Skype or, Zoom or whatever. We have to use that online tool. But yeah, sometimes I'm very uncomfortable in my apartment. So it's hard to say. I cannot say anything about, our lab. So yeah.
I think that's a great point that Yoshi brings up, though. And so maybe as we move through the conversation, it really is a reflection on how COVID has impacted things. Maria?
Yep. Yes, I actually agree a lot with Yoshi. And because I spent my first 9 months here in Calgary in my apartment. I could not visit anything or meet the other lab members. And it was quite difficult. And it actually relates with the health issue that we were talking about in the previous section, I guess. That it helped me a lot when I was able to go again back to the lab and meet all the other people and actually have this small talk and how was your day and these things. But now things are getting better and hopefully I have more opportunities to meet, to know Calgary and what it has to offer.
Yeah, fortunately, COVID did not impact us that much because we were allowed to go work in the lab almost every day since we work with mice. And just like little babies, they need constant nurturing. Like you need to take care of them, see if they're doing well. And yeah, I would like to say that research is not just research, it's a journey. And yeah, we try to enjoy every day, even though 90% of the times we just experience failures in our experiments. But I think even if one day is successful, that brightens up the rest of the month for us. Yeah, that's it.
Yeah, I think. Yeah, for me, I think it's mostly I have the impact of COVID on my research too, because usually when you study cell process, you have a wide range of techniques that you could use. You go from studying patients in the hospital to looking into their blood, studying proteins, studying genetics, using imaging, MRI, ultrasound to see their arteries, and also the statistics, like getting large data sets and trying to compute models. So you can see that the aspect, the clinical aspect, like imaging that we had planned to do, is not visible in what I've presented, mostly because the MRI unit was shut down due to COVID, so I had to find another way to still move forward. So this is how I got the data set from the US to continue doing the analysis.
So interesting. And Tejal, you brought up an excellent point too, where 90% of your work as researchers is failures or lessons learned, I would say. And that's similar in terms of the entrepreneurship space and innovation space, right? To innovate, we're going to fail 90% of the time. We're only going to be successful 10% of the time. But those successes are invaluable to the progress of our community. So I applaud each and every one of you. You guys are doing some fantastic work. Postdocs and trainees are such an integral part of the university community. And the work you're doing is so important. And we're excited to see the results of it. Thank you so much for joining and sharing your time with us today.
Thank you.
Thank you.
Thanks, everyone.
Thank you.
We'll be back in about 5 minutes with our session 4.
Thank you.